Good evening, I'm Curtis Aaron, and this is Supreme News Network with a special report breaking tonight on scientific advances in the conformation of reversible DNA I motif structure. Sound confusing yet? We'll explain more later tonight on our Science News special. Now we cut to our lead correspondent, Varun Kataria.
Good evening, Curtis. We are here in LA with our lead researcher in the Royal Society of Chemistry DNA department. So Henry, before we get into the specifics of what your team found, could you explain to us a little bit about the polymorphic nature of DNA for our viewers at home who aren't too familiar?
First of all, thank you for having me on the show, Varun. For background, when working with DNA, different types of secondary structures are formed. These structures are based upon a sequence of bases, the pH level, and are influenced by environmental conditions. I-motifs are four-strand nucleic acid secondary structures which require folding to function properly. Typically, the pH is changed to fold these structures, but in our experiment, we hypothesized certain cations would be able to affect the I-motif structures.
Okay, and prior to your team conducting the experiment, has anything similar been attempted? What sets your experiment apart?
Previous experiments have been completed regarding pH and pH-driven nanomachines to cause folding. These have been completed with certain cations such as silver, which allowed us to hypothesize that other cations could influence structure and function.
And from what I'm familiar with, copper cations were used in the experiment?
Yes, the use of copper-2 cations were utilized to fold DNA into the hairpin structure. If DNA was stretched out, it would be extremely long and unmanageable. To combat that, DNA must be manipulated into a manageable size so that it can fit into the nucleus of a cell. To do this, the DNA must fold, which is almost exactly what it sounds like.
And what is the benefit of this hairpin folded structure?
The hairpin structure is vital to having more extensive applications of DNA in creating structures in biology or nano devices. Our discovery is groundbreaking that we are able to fold DNA in a way that we were unable to do previously.
How were you able to indicate the type of folded structure through your study?
We were able to confirm the folding effects of the copper cations by using UV difference spectroscopy. Different structures of DNA absorb nanometric wavelengths of UV light depending on whether they are folded or unfolded. We are able to have a larger range to determine whether the result of the cation exposure was successfully folded. Another type of spectroscopy used was circular dichroism. The main purpose of using two types of spectroscopy was to ensure that collected data could be analyzed correctly and accurately, something that is very important in our research.
Now, is there a way to perhaps unfold this DNA if necessary? Have any solutions been found to this problem?
Yes, actually another aspect that my team and I worked on was another way to unfold the DNA without changing the pH.
Oh, so could you explain to us a little bit about the purpose of that experiment?
So it's important to us to examine the reversibility of our previous process. Our team uses an acid called EDTA, which can be used in an advanced process known as chelating, which can reverse the effects of copper-2 ions in the DNA. As such, we analyze the effect of adding EDTA in after titration with copper-2 plus ions using circular dichroism spectroscopy, which we talked about earlier.
Wow, that's fascinating. Wow! Fascinating work, Henry, and my sincere congratulations to your team for such a great scientific advancement. Thank you for coming in today and explaining its significance.
Not a problem. It was my pleasure.
Now, back to Curtis with the weather update.
Thank you, Varun. It's hot here. Wear shorts or something.
It's still alive. It's dead. It's not so bad.
That John's still alive.
I-motifs are four-strand nucleic acid secondary structures which require bases and are influenced by environmental conditions. I-motifs are four-strand nucleic.
Okay.
Give me a minute. Okay. So Henry, before we get
sorry.
yeah, ready?
Alright, okay. Alright. A little bit about the polymorph. Jesus.
You can even hear that.
Yeah, okay, fine.
Which require folding to function properly. That's an inside joke we have in our team.
Go!
Don't do that.
3, 2, 1.
Okay. See, what happens is Varun will do like these subtle things, and he knows.
I gotta do something. I just look so weird. And what is the benefit of this hairpin folded structure?
The hairpin structure is vital to having more extensive applications of DNA in creating structures in biology or nano devices. So, our discovery is groundbreaking that we are able to fold DNA in a way that we were unable to do previously.
Alright, no, come back. Yeah.
Alright.
3, 2, 1.
You guys just think you're
I can't do it. I
think you're cute. No.
Alright, stop. Chill.
Yeah.
Wow, fascinating work.
Go.
Wow, fascinating work. That was funny. All right, we can do that though. You have to be very.
Right.
It's too set up. All right, stop.